Years ago, you could forgive someone for thinking that machine learning was solely the play space of Silicon Valley technologists. But since the use of artificial intelligence and its closely connected sibling, machine learning, have spread across disciplines, including medicine, in the last decade, it's impossible to suggest it belongs in any one professional corner. So much so that one day in the not so distant future, women's health doctors, like OBGYNs, may be using meticulously honed prediction algorithms from the March of Dimes Prematurity Research Center at Stanford to make vital decisions about the treatment of preterm birth and preeclampsia in their patients. And that, my next guest, Dr. Brice Gaudillière, says is a good thing. In fact, he argues, it brings us into the realm of precision medicine, the holy grail of medical care where each patient is treated based on their unique history and biology. This new world of precision medicine has only become a reality in the recent past. I'm talking about January 2024, when the scientific outlet Nature Biotechnology published Dr. Gaudillière's paper on a new machine learning model that had managed to do the impossible. To unlock the so-called black box of predictive AI algorithms and identify the biological causes behind those predictions. In other words, if previous AI algorithms could identify pregnant women at risk of preeclampsia, this new model could do the same, plus something else very important. It could also, and this is where other models reach their limit, provide a short list of a dozen or so biomarkers or biological elements like proteins or genes that triggered the risk prediction for that particular woman. So it was providing the evidence behind these predictions. In the hands of a doctor, this biomarker information would be invaluable. It would allow doctors to treat the exact biological cause of a particular medical issue, providing targeted medications and interventions instead of blanket solutions that don't work for everybody. And there's more. Knowing which biological causes are linked with which pregnancy conditions allows experts to work on creating tests for preterm birth and preeclampsia and searching for existing and new medications to treat these conditions. And it's all because of biomarker identification. It's why this machine learning model is so unique, so important, and so very interesting. So let's get into all the details with Dr. Gaudillière, the lead author of this biomarker paper and one of the key players behind the prospect of making precision medicine a reality for pregnant women.
The views expressed on this podcast may not represent those of March of Dimes or its staff. Information presented on this podcast is not medical advice and should not be construed as such. When in doubt, seek competent medical advice. Now, on with the show.
Dr. Brice Gaudillière is an associate professor of anesthesiology, perioperative and pain medicine, and of pediatrics at Stanford Medicine. He is also an investigator at the March of Dimes PRC at Stanford and led the biomarker study as part of that team. Brice, welcome to the show.
Well, my pleasure. Really happy to be here.
Happy to have you. As you may have guessed by my pronunciation of your name, I tried very hard, but I'm not French, but you are. How did you come to be at Stanford and how did you come to be in this field?
A bit of a windy road, actually. And by the way, your pronunciation is on point. Much better than average.
Thank you.
So I studied, I'm a product of the French engineering school, so meaning I lots of math and theoretical physics and chemistry as a background. Then I was lucky enough to come to the US for a PhD in biomedical sciences and then an MD training at the interface between engineering and medicine. It was a program between Harvard and MIT called Health Science and Technology. So a way to kind of keep that engineering background in the medical studies. Then I came to Stanford, specialized in anesthesiology, and then really a turning point in my career was I joined a systems immunology lab, Gary Nolan's lab, at Stanford for a postdoc at the time where new technologies were being developed that really revolutionized immunology or modern immunology. One of them called CyTOF or mass cytometry really allows to map the immune system with very high resolution. And my job when I started the lab was really to bring these technologies to the bedside and answer clinical questions. And that's when David Stevenson, who's the PI, the director of the Stanford Prematurity Research Center, the March of Dimes Prematurity Research Center, I think he heard about what we were doing and brought us to the team and kind of see if it made sense to apply these technology in the context of maternal health. And that was a really fantastic opportunity to study and use these technologies to understand maybe the most fascinating immunological paradox in human biology, which is pregnancy. And that's what we've been doing for the past decade or so, 12 years. So understanding immunological mechanisms of healthy pregnancy and identifying predictive biomarkers of abnormal or pregnancy complications like preterm birth and preeclampsia.
So before that decade started and Stevenson asked you to come on board, were you involved in maternal immunology at all, or was that you called it a turning point? But where would your career have gone if he had not knocked on your door, do you think?
Yeah, I think the lab is really interesting in how the human immune system responds to stressors in general. I'm an anesthesiologist, so we work in surgery and traumatic injury. Surgical injury is a massive stressor to the immune system, and this response to stress is extremely important in understanding clinical outcomes in this context. Well, pregnancy is another stressor. You have your immune system at baseline, and then all of a sudden there's a baby, a fetus that's half the genes are coming from the dad, so not supposed to be compatible with the maternal immune system, and somehow the maternal immune system needs to maintain tolerance and deal with this otherwise foreign thing. And so that's how I got interested in fetal maternal immunology. Really through that collaboration.
Wonderful. So we owe a big thanks to David Stevenson then.
Absolutely.
So you're an anesthesiologist. You practice
Yes,
practice anesthesiology on which patients in particular?
All patients, and I'll tell you a little story because, and that's the truth. At the time where David was recruiting me into the center, I was actually doing a lot of obstetric anesthesiology, and so that's where the anesthesiologists also have like a very specific niche. Taking care of pregnant mothers or about to be mothers at the time of labor, a really intense time.
And most of that, Brice, is epidurals and cesarean.
That's that, yeah. So epidurals is kind of like the bread and butter, and then dealing with very quick paced intervention that need to happen when things go badly in the context of labor, and that's where the anesthesiologist and the obstetrician are really a team in things that go really where you have to go very fast. And so, so there is, an immediate and very logical connection between these two fields. And so all this was coming together and made a lot of sense at this time in my career.
Wonderful. Well, we're so glad that you are here. So the big topic of this episode is precision medicine. I touched on what that is a little bit, but what is it exactly in your words? So if you were in an elevator with somebody and you said, the things that I'm working on could help make precision medicine possible one day. And they said, what's that?
Yeah. And I think you started very well at the beginning in your introduction. And to me, it's really a situation where the care team or the physician is using information about the individual patient in order to change the management and improve a certain outcomes, a certain disease process, certain outcome for this patient. And the key term here is individual information as opposed to population-based information or average information. This, in order for precision medicine to happen, you need to have information that is predictive or associated with a certain outcome that's accurate enough so that you're really able to take into account all the biological, physiological, demographic parameters, information, the microbiome, the immune system, the metabolome, that really makes this patient an individual, and take this into account in order to make an accurate prediction. So that's really where the precision happens. And yeah.
And that's not at all what happens today, is it? Especially in the field of pregnancy, everyone is kind of, I mean, obviously some women will have gestational diabetes and be treated for that. Others will not. Others will have preeclampsia, et cetera. But there's kind of a toolbox that doctors have, and women, fall in various parts of that toolbox. It's not as personalized as I think women would like.
Yeah.
And birthing people.
Sure. I mean, I would qualify this a little bit in the sense that, in a way, precision medicine is how medicine has been performed, forever, ever since, people were taking care of pregnant women or patients in general. If you think of someone coming to your doctor because of, high blood pressure, a primary care doctor, you're going to measure the blood pressure, then a couple months later, it comes back. Is the blood pressure higher or lower? And then you devise, you change the blood pressure medication regimen based on that. That's precision medicine. I think what has happened, with the advance of modern epidemiology, with Claude Bernard, et cetera, is that there is a push and a revolution that happened in terms of evidence-based medicine that has, really, we've heard about this in the last 3 decades, really applying knowledge and evidence that is gathered based on understanding and measuring data at the population level, so on many patients, and then using this information to then change or advanced medicine, change patient management, and certainly in fetal-maternal medicine, that evidence-based medicine is really important, and that has helped the field immensely. But now we're in the next phase of this evolution, where clearly we are more and more understanding that the data that's gathered at the population level is not representative of individual people. So, for example, in this the problem that we study, which is one of the problems we study, which is a prediction of onset of labor, prediction of the due date. Well, if you're taking the clinical tools that obstetricians use, right, the definition of onset of labor or due dates is at 40 weeks of pregnancy, and obviously, anywhere between 37 and 42 weeks, this is all healthy labor, and depending on the individual, 40 weeks doesn't mean anything for that particular patient. Particularly if you're at risk of preterm birth, then you're completely off the chart. And so this is really the difference between population and individual information. And so in the last 10 years or so, there's really been many studies that are pushing the needle towards precision medicine, and for this is really enabled by new technologies that can allow us to measure biology, measure mechanisms that are linked to these pregnancy outcomes, and understand this with really fine resolution. So basically, understanding the differences in pregnant mother microbiome, immune system, metabolome, cardiovascular system, etc., with extreme resolution, so that you can really understand at the individual level what makes a pregnancy different from the other. And to summarize all this, it really, at the end of the day, it all has to do with identifying biomarkers. So now that we can measure thousands of parameters, thousands of omics parameters, the task has become, well, which one of these parameters that are so precisely defining an individual person are really related to the outcome? Which one are really predictive of the outcome? Which one are the actual clinical biomarkers? And that's where we are, and that's the difficult task that we have to really bring precision medicine to the field of fetal maternal medicine.
Right. And this is what this is why this model that we're talking about today is so important, because it helps bring us from a very large, potentially arbitrary list of biomarkers to a much smaller, much more trustworthy list of biomarkers that are actually much more likely to be related to the specific prediction. And so this all has to do with this model, this machine learning model, in the greater story of artificial intelligence. And so I'm certainly no expert. So maybe you can kind of set the stage here. When people talk about finding the needle in the haystack, does that mean separating the signal from the noise? Or does that mean finding the biomarker that's responsible? Or does it mean both? And can you just talk a little bit about what noise is and what signal is, and just in general, this kind of set a foundation for us?
Yeah, absolutely. So, well, let's start with, what is noise, what is signal? In our language, really, signal means a parameter that's being measured, let's say in the blood, that is truly predictive or associated with an outcome, let's say preterm birth. Okay, noise are parameters that are also measured at the same time, right? But they're not associated with this outcome, truly associated. They may, by chance, because of the actual the population that you're studying that day. But if you were to measure every patient in the world, then you would see that they're not associated, or not informative. So noise is non-informative parameters.
So it's kind of like a multiple choice test, right? A, B, C, D. If the answer is B, that is the signal, and A, C, and D is noise.
Is the noise? It's trying to
the wrong answer, the potentially,
you.
Potentially the right ansers, but the wrong answers ultimately.
Exactly, and they're all there, right?
Yeah.
And when you're taking the test, you don't know
right
until you know that you were right or wrong. You don't know what the signal was, and I mean, in a sense, this is what many machine learning methods is all about, is how do you distinguish signal from noise. If you take hear a lot of self-driving cars these days, and image recognition, right? Trying to distinguish between a flying ball and a flying person in front of the car, that's a life and death decision. How do you find the detail in these images that are allowing this algorithm to really make the difference between these two very different objects, right? And so for this, the process is that these algorithms are fed a bunch of data, okay, where what the outcome is, the difference between the ball and the person, and then the machine learning algorithm is learning, it's training to identify what are the components of this image, let's say, that allow to make this prediction. And this is really the learning process. And then there's a second process that's very important, which is once you have the model, once you think you know how to make this prediction, then you're testing this model on a new image, on a new patient, on a new set of patients, and see how good you are and how accurate you are.
Mm-hmm. And I mentioned earlier the difference between your model and previous models, some of which you worked on, probably.
Mm-hmm.
The entire industry has been working on the old standard before stable, and that is the name of your model, or this new technique is stable. And previously, at least in the field of maternal health and immunology, like what you're studying, we're talking about something like 200 biomarkers. A model would identify 200 biomarkers, or some number similar to this, a very large number. And what you told me in the past is that actually every time you run the model on the same data set, it would give you a different answer of 200. A different bundle of 200 biomarkers would come. And so that was probably pretty frustrating. And so you said, you know what, let's make something better. We need to lower that number, bring it down, and make it more reliable. And so that's what you guys did, you and your team. And specifically, there were 3 conditions that your model made predictions on. We talked about labor onset. So one of them was labor onset. When will a woman go into birth? Microbiome driven preterm birth. So early labor that happens as a result of the microbiota in a woman's vaginal canal, and preeclampsia, the most dangerous hypertensive disorder of pregnancy. And it shortlisted 20 biomarkers for labor onset, 12 for preterm birth, and 9 for preeclampsia. I mean, that, 20, 12, and 9. I can memorize those, It's such a small number. So congratulations.
Yeah, so that was really what started this whole project here, was a limitation in the tools that we had to find these biomarkers. And it's I'll talk about it in a few minutes, because it's important for this field of pregnancy outcomes, but also for the field of using these omics data sets and these omics technologies to find biomarkers in general, and that really what started this whole project. So there's something called the curse of dimensionality that I think you mentioned in your introduction, right? Which
I didn't, but please talk about it. I was going to talk about it later, but talk about it.
Yeah, so it's a statistical problem that we have to face. Basically, that says that if you have the more parameters, the more features you have to describe a system, or to describe an outcome, or to differentiate two patient population, the more things you measure, the more samples, the more number of patients you need, not the less samples, the more you need, right? And that's a problem in our field, because we're very good at measuring many, many features now. If you take transcriptomic data sets like cell-free RNA, we're measuring 20,000 features. In mass cytometry, measuring thousands of features, in metabolites, thousands. But these technologies,
and that's from what, a single drop of blood from a single person.
From a single person,
Yeah.
From a single blood, we're measuring thousands of features.
Wow.
But these technologies are expensive. These technologies are not, you're basically limited in the number of patients that you can recruit in these studies. So usually we have maybe 100, typically 100, a little less than 100, very rarely thousands of patients, but definitely not, the same, definitely limited in the number of patients that we have. And so when you have, in this context, right, doing a prediction and then identifying what is signal from noise becomes statistically very difficult. And there's a whole field of machine learning that was really started by Raptip Shirani and Trevor Hasty at Stanford. A set of machine learning algorithm called regularized linear regression models that really get to this point. How do you build a predictive model and then identify these biomarker the most accurately possible? But even using these existing algorithm in practice, and there is a known limitation with these algorithms, is that if you are running them on one data set and then change that data set a little bit and run them exactly again, then the biomarkers are going to be selected very widely. And even though these machine learning algorithms help in reducing this number of biomarkers, you still typically end up with way too many biomarkers to make these predictions. And why is this a problem? Well, first of all, it's a problem when you're trying to do the clinical translation. So it's really nice to have a prediction, and an accurate prediction saying, I know with 90% chance that this woman will have a preterm labor or not. But if you're trying to develop a clinical diagnostic test, right, you also want to know, how am I doing this prediction? What are the 5-10 biomarkers that makes this prediction? It's going to be very difficult to develop a clinical assay with 100, 200, or 1,000 biomarkers. You're going to need to really reduce the number of biomarkers to develop a clinical test. Similarly, if you want to develop a drug target or identify new drug targets.
Is that because of cost? Cost? You want to bring it down, or?
The current state of the clinical grade instruments, the clinical technology that can be really scalable so that you can deliver this diagnostic test to millions of patients is such that, to date, we can't measure hundreds of parameters. Okay.
So going back to the old way, the 200 versus the new
Yeah.
way, let's say 12, is we want to go, obviously we want to shrink that number because you want a device that can be used in the clinic, can be scalable, like you just said. But is it also true that those 200 were just not accurate and that actually only a smaller number is more accurate? Or is it true that no, all 200 of those are biomarkers that are related to the prediction?
Yeah.
Yeah,
So
which is it?
typically, yeah. So typically, what would happen is that within these 200, and again 200 that change when you run the algorithm again and again,
Yes, that's
many
not
of these
helpful at all.
biomarkers would be selected that are actual noise.
Okay.
Okay, they
So
would be
there was a lot
selected
of
by
noise.
chance.
Yeah. Okay.
And
Yes.
so that threshold of signal to noise is not obvious. And that was really, the defining question and the defining problem that set us to come up with a new algorithm. We needed something better to not just make prediction, but also have an objective way, a data-driven way that says, okay, those biomarkers, there's a real signal. If I measure them again, they're going to be the ones that I'm going to find. They're reliable versus these other biomarkers. Well, they kind of got into this model because this is the data that I have, but really they're noisy. And, they're, shouldn't pursue them.
Yeah.
Yeah.
So then let's talk about how Stable works. How was it able to do that impressive editing or shrinking job? When you talk to me, I've talked to you about this topic multiple times, and you basically the way that your team did it is you guys injected noise. You guys injected fake noise. You created a data set of fake parameters. You had a lot of noise in there. Or not parameters, but yeah, all sorts of parameters. And then you put it on top of the real noise in the real data set. And then what happened?
Yeah, so that was the cool moment. So Julien Edu, who is the first author, who is also an engineering student, was reading the works of Emmanuel Candes and Andrea Montanari, who are specialized in noise, in the field of how do you model noise. And he had this great idea of using a similar method with the thought of, well, if we can't really know what is noise in our data set, what if we were building another data set that looks very similar to the data set that we have, except that we know that every parameter in this data set is noise, meaning it's not informative. It's by design not related to the outcome that we're studying, like preterm birth. And then we mix the two data sets. So we mix the real data set and the noise synthetic, fake data set together and start running our algorithm on both data sets together. That way we can train the algorithm in the training phase, right? We can, whenever the algorithm is selecting a fake synthetic noise, it will know for sure this is noise. And that allows,
because you tell the machine that it's noise, right?
Because we know it is, right? We know for sure we made it. By design, this is noise. And so the machine will learn what does noise look like. We'll apply that to the real data. This is, okay, this other biomarker, a real biomarker, looks like noise, and will, in an objective way, in a data-driven way, will set a threshold that says, okay, this biomarker is signal from noise.
Can I ask a question? So how does it, I know that it learns, right? It's iterative, it learns, but how exactly does fake data look like real data? Is that how it learns? It sees like, okay, fake data looks like this. I'm almost seeing like a molecule, right? Okay, this is fake data. Oh, that looks similar. That must be real. I'm sorry. This is fake noise. Over here, it looks similar to that. That must be real noise. Is that how it learns?
A little bit. And certainly it's important that the noise looks as close as possible to the real data. Okay. And technically the way it works is really when the machine learning is learning, we are running and building a model on both data set, then changing the data sets a little bit, running it again, etc., 1,000 times. Every time that a biomarker, whether it's real or fake, is selected, we count it. And then you can count the frequency at which a real biomarker or a noisy biomarker or a fake biomarker is actually being selected. And that allows you to say, well, this is the threshold, this is the frequency threshold of noise. And anything that's being selected at a higher frequency, that's real, that's the real biomarker, that's the signal. The key, an important part here is that the user, the statistician or the investigator doesn't have to intervene. We're not saying, oh, I want a p-value of 0.01, or I want some sort of subjective way to say this is good, this is bad, this is higher or lower. It's really driven by the data, and this threshold is driven by the actual data set that we're using, and that's why it is a very robust way to define that. And this work, was done in close collaboration with Andrea Montanari and Ivana Maric, who are specialized in these, from Andrea in a different field, who are specialized in kind of using this type of method for to generate noise and to model noise and data.
Was it a completely novel and unique original idea to create a fake data set and have the model learn from the fake data, from the fake noise, to identify real noise, or was that something that had been done in the past? Maybe not successfully, but
no, it was. I think it was quite novel in our field, right? In other field, it's always, you always learn from other fields, and you're like, okay, well, that looks like it's gonna, it could work, and then it turned out to be really a key to open that solution in our field.
And you're talking about your field, but this model can be applicable to many different fields, right? And it is open source, which means professionals in other fields that use data and are looking for their own needles in haystacks can use it. And you have been contacted by various other folks in different industries. Can you talk a little bit about what another good use case of this model would be, and who has reached out to you?
Yeah. So as I said, this we tested the algorithm on multiple types of data set clinical outcomes, not just in female health. And so we got a lot of feedback and a lot of interest by different groups, whether it's in academic centers. So we have a collaboration to study outcomes after transplantation, outcomes after stroke, cancer, and as well with industry, with for example, Genentech and Novartis are interested. And so I think the solution that we're providing is practical enough and tangible enough that you can apply it to many different fields. We have a collaboration to personalize or do a biomarker-driven clinical trials in the context of cancer using this method. We were contacted by NASA also. We're talking to them next week. We'll see. That's a super exciting opportunity, because guess what? When you're an astronaut, you go to space, there's going to be a lot of experiments that are performed, and we're going to measure a lot of things in your blood to understand, the biology of space and what does space do to your immune system and other systems. And there are not that many astronauts. There's not like thousands of astronauts that you can sample from. And so that's exactly the type of question that we have. We're going to measure so many parameters, but just, dozens of observations.
So
that's super exciting.
That's really cool. What would be what is the question that they're trying to answer at NASA? They're trying to say, how does space change specific biomarkers, or what biomarkers in the body are changed as a result of space travel?
Yeah, I mean, there's a huge
life.
I mean, those are decades worth of interest from NASA to understand the effect of long-term space flight on physiology and health issues. So there's bone loss, there's changes in circadian rhythm, there's infectious problem, etc. And basically, how do you tune up humans to be able to withstand all the rigors and the difficulties of long-term space flight? super exciting stuff, and I can talk about
Yeah,
this for a long time. It's
absolutely.
a bit deviating from maternal health,
Yeah,
but
it's a little bit of a tangent, but just to close that conversation, it seems like space would affect every biomarker in your entire body, which is kind of the opposite of your model, right? Your model is to shrink the number of things that are influencing, to be very specific with what is being influenced. But it seems like being in in a shuttle, everything would be impacted. Of course, I'm not sure if that's true, but
well, what? So no, I would say the opposite. I would say, you
yeah,
have space, zero gravity, radiation, etc. Those are massive stressors on your
yes,
physiology, right? You want to know exactly what are the biomarkers that are related to the effect of these stressors on an outcome. Let's say you get an infection while you're in space, right? And you need to know exactly which one are important from which one are noise in order to develop countermeasures to follow over time people, and it's the same idea. You can't keep measuring everything on everybody at all time. You have to at some point narrow it down to what is relevant, what is informative.
right. So the question is not how does space affect the body. The question is there's an infection of the spleen or something as a result of something that happened, and what are the biomarkers associated with that particular
Yeah,
condition
yeah, so that
while
you
in
can treat it.
space?
Yeah,
Yes,
yeah,
treat it.
exactly,
Yeah,
so you can intervene on it.
yeah, yeah. Treat it and somehow change the human in order to be able to better withstand it.
Exactly.
Okay. Well, wow.
The next
Interesting.
frontier.
That's right. Congratulations to you on that meeting next week or whenever it is. I hope it goes really well. How long did it take you all to complete the model? When did you guys start working on it until January the paper came out? So how many years was this in the making?
Yes, yeah. Between the time where Julian and Julian started thinking about it, and working with Ivana and Andrea, I think it was good 3 years of developing the for the first 2 years. It was really developing the math and the algorithm, testing it on synthetic data, having a mathematical proof around why it's working so well. And then it was just, okay, let's now apply it to real world data.
Yeah.
And a couple of these data sets we generated ourselves, so that takes time as well. We didn't want to just use data sets that were available publicly, although we did in many instances, but we also wanted, in a new data set, how does that perform? And so all this took about 3 years.
And obviously, you're looking to evolve the model and make changes and to infinity and beyond with this model, literally. One of the changes that you're looking to enact is you want the model to be able to work with clustered data or clustered biomarkers. Can you talk about what those are?
Yeah.
Um,
Yeah, that's definitely, so that's a line of future directions. I mean, it is already working pretty well with clustered data, but there's more to be done. And so this algorithm is a framework. You can, there's building blocks. There's how do you make the noise. There's what is the algorithm that you use to make the prediction, and all this you can kind of modulate it based on what the needs of the data set. The way that we model noise has mathematical limitations, and there are actually it's a whole field of math around this, and there's definitely some improvement to really be able to make noise look exactly like real data, with all the the correlation and the interaction between different features and data. All this, there's improvement that needs to be made to really be close to the real data set. Another improvement is in the machine learning that we're using to make predictions. So we're using linear models. Obviously, medical data, biological data is not linear necessarily. So you can think about nonlinear approaches to improve on this, and other direction like that to improve the accuracy and the biomarker and the identification eventually.
right. So all of so far we've been talking about what these models, what state these models can bring us to, a state of precision medicine,
Mm-hmm.
the work behind the model, how the model came to be. but not the most interesting part yet, which is what did the model tell us, right? So let's move into that part of the discussion. You told me something I really loved on a background call I had with you. You said that one of the reasons that you guys did this, and one of the values of this model, and all models ideally, hopefully all models go in this direction, is to give scientists that word, direction, a place to go, a new path to pursue, where there are more answers. And from the biomarkers this model identified for labor onset, microbiome-driven preterm birth, and preeclampsia, these biomarkers include genes, proteins, immune cells, RNAs, metabolites. You mentioned omics. This is together known as omics. And there was one really promising biomarker that you have identified as kind of the most interesting lead for you all. And that was for the predictions of preterm birth and labor onset. And so if there was one thing that came out of these three models and predictive models and the biomarkers that resulted, if there was one, it would be this one. And we'll talk about that in length, but I just wanted to know, and those were for labor onset and preterm birth, but I just wanted to talk to you about preeclampsia for a second. Nothing super interesting there that you're pursuing with preeclampsia for the biomarkers that were identified?
Plenty.
Plenty. Okay.
Yeah,
okay.
Absolutely. So the preeclampsia data set, some of the application is stable in the paper are cell-free RNA data set that was generated by Mira Mufaraj, that I think you interviewed before.
Yes,
Steve Quake,
yes.
Another one.
A rising star in preeclampsia research.
Absolutely.
Yes.
The microbiome data set was from UCSF with Marina Sirota. But staying in preeclampsia from this work and then the work that we're doing with Virginia Nguyen and Nardi Gomez-Lopez, and you know all these guys, stable was really useful to hone in on specific pathways that have to do with, in early preeclampsia, how the maternal immune system falls off from mechanisms of fetal-maternal tolerance to inflammation. So there's specific cells called Th17 and Tregs that kind of really fine-tune the balance between tolerance and inflammation. And some of the biomarkers really have to do with this balance between these two cell types early in pregnancy. So very important line of research that we're pursuing with other data sets, longitudinal data sets that we're doing in this context. I think are very promising. And then in terms of the onset of labor, that it was really useful and really kind of like jump.
Can I interrupt for a sec, Brice?
Yeah,
Sorry,
yeah.
I'm just thinking about the preeclampsia one that you just talked about. So there's an important balance between these different cells. So where would that lie? What is the next step in that work? I mean, ultimately, I guess, what would be the last step in order? Would you want to introduce more of one particular type of cell in the maternal blood, or facilitate the growth of more of these cells so that or less of another, in order to have this balance so that preeclampsia has less of a chance to take hold in the woman's body?
Yeah, what the data is showing us is how these regulatory T cells, how functionally they become stable and stable at the time of pregnancy, where they're so important. At the beginning of pregnancy, there's the initiation of fetal maternal tolerance, and if it goes wrong, it is associated with abnormal placentation and development of placenta that leads to preeclampsia. So at that time, if you could modulate the signaling that is coming out of the model, right, using various pharmacological intervention to foster the stability of these cells at that time, then you could think of new ways to pharmacologically prevent preeclampsia.
Well, that's really big. That's big.
At the end of the I mean, there's many studies and a lot of development towards that, but it gives you a path.
Are you looking at
research.
Are you looking at drug repurposing work with for that too? How
Yeah,
to
yeah, we can talk about that. Yeah,
on a
definitely.
separate podcast? Yeah,
Yeah,
wonderful.
yeah.
That's great. Okay, so this particular biomarker that so that was for preeclampsia. Now we're going to talk about preterm birth and labor onset, and that biomarker that interested you guys. It is an inflammatory cytokine, or a protein called IL-33. Tell me about this guy. This guy's a precursor to labor, right?
So yeah, so IL-33, there is more and more evidence, predominantly from animal studies, from mouse models, Adrian Arlebacker's work, particularly at Stanford, that shows that it is absolutely important in inflammatory mechanisms that trigger labor.
Mm-hmm.
And those mechanisms are at the fetal maternal interface, okay, the local mechanisms that are important in the trigger of labor in these animal models. When we run our algorithm for the prediction of onset of labor, both in term and preterm pregnancy, the top feature is related to IL-33. It's not IL-33, it's IL-33, an inhibitory receptor of IL-33. So first of all, it was super exciting that, completely agnostically, this is what comes out, because we know that the biology makes sense locally. So is it, are we really finding the ripple effect of local mechanisms that are triggering labor? And we're picking up on this biomarker in the blood. That's very satisfying. But as you know, these models it's not just this receptor, circulating in the plasma. The predictive model is this biomarker in conjunction with a dampening of inflammatory response that are measured at the single cell level, and that was surprising. So, from a biological standpoint, right, it really helps understanding what the biology of labor that is linked to the onset of labor, as if at the same time as you have these pro-inflammatory mechanisms that are necessary for the onset of labor, systemically, the peripheral immune system, it's as if the mother knows that you have to kind of control this system in a negative feedback loop, and make sure that the inflammation doesn't go awry, and everything is kind of like a gas and a throttle type of situation, where you can control this inflammation and have the birth of a healthy baby. That's one interpretation. But it was really exciting when you find a few set of biomarkers that are predictive of such outcome, and that makes sense when you look at them all together, and it kind of
Yeah.
paints a biological picture that you can follow, and you can think about intervention that would target these specific pathways and these specific mechanisms for changing the time of labor, basically accelerating or decelerating it, depending if you're preterm or postterm.
Yes, exactly. Does this have to do with are these monocytes that the protein that the receptor exists for? Is it a monocyte? Because I learned something from our recent Basil O'Connor Award winner,
Yeah.
a woman who's working at Mayo Clinic at Minnesota, a doctor, and her work is on cell-free fetal DNA, and she says that the more that towards the end of labor, cell-free fetal DNA really increases in the maternal blood. Something like 10% of the maternal blood is cell-free fetal DNA, and this is what and it triggers monocytes to ramp up their immune response, which leads to
Yeah.
labor. And I wonder if IL-33 is a key player in the same process.
Yeah, and so monocytes in pregnancy, super important, and it's that's why you need single cell resolution. So there's monocyte and monocyte. There are the monocytes that are driving inflammation, okay, maybe in response to IL-33, that are important in labor onset. At the same time, there's anti-inflammatory monocytes called myeloid-derived suppressor cells, also monocytes, and they're also important in pregnancy. And so that's why you need to have a technology that allows you to differentiate all these different subset of cells. In our model, it's the inflammatory type that come out, that seems to be kind of dampened, negatively regulated as the mother approaches the time of labor, at the same time as when, locally likely it's inflammation that is happening that triggers labor.
And you mentioned a couple minutes ago that having all this information in hand would allow you to find maybe existing medications or create new molecules that could, depending on the woman's gestation and when she needs to deliver, when is healthiest for her to deliver, either speed up or slow down gestation. So let's talk about a little bit about what the next steps of IL-33 are for you and your team, and in trying to find a medication that can affect this receptor, but not affect anything else in the process that initiates labor.
Yeah, exactly. I'm glad you're mentioning not affecting anything else, because that's the key, and that's kind of driving that pretty exciting line of research in the lab. So to start with, IL-33 is a master regulator of inflammation. It can modulate monocyte and drive inflammation through activating monocytes, but it also activates and regulates regulatory T cells, these anti-inflammatory cells. Okay, so IL-33 can do many things. And so in the drug discovery process, right, you want to be able to select, to identify new drugs or existing drugs that have selective properties. You would want to kind of a drug that dampens inflammation, IL-33 mediated inflammation, but that not touch necessarily the role of IL-33 in Tregs and in the maintenance of tolerance, for example. That would be really, really important. And so that's why we're taking a bit of a new approach to do drug discovery in this context, in collaboration with Marina Sirota. A bit of an unconventional approach, a drug repurposing approach, where we're starting with all the different FDA-approved drugs that are being selected through the screen, the drug repurposing screen that she has developed. Okay, so that gives us a short list of drugs that are potential candidates for preventing preterm birth. But if you think about it, these drugs, their properties have been defined using data generated in cancer cell line. It has nothing to do with real primary human tissue, let alone immunological tissue or immune cells. And so in a high throughput drug repurposing system that we've developed, what we've done in the lab is to develop an assay that allows testing the effect of all these drugs, not only on the mechanisms that would be the right target, let's say IL-33 mediated pathways, but at the same time monitor the effect of these drugs on the entire immune system in the context of the pregnancy immune response. Okay, so to identify selective drugs that has a beneficial effect, a selective effect, while avoiding potential side effect on the other branches of the immune system. And now we can do this, with very, very small amount of blood, like 100 microliter of blood, and screen through thousands of different immunological responses, and at the same time, right, simultaneously define what is the effect of these drugs on the entire immune system. So it's as if we are creating a digital twin of the maternal immune system using this assay, and then test how these drugs is going to modify this digital twin and get a precise sense of the effect on the whole system and on the outcome that we're interested in. So that then we can say, okay, well, this drug looks really promising, not only because it decelerates the time to labor in the context of preterm birth, but also it's not affecting, immune cell response that it shouldn't be affecting.
What's a good example of the perfect type of drug, not by name, but by what it would do? So like what you just said, it is slowing down preterm birth, but it is not causing negative immune effects. What's an example of a negative immune effect that could happen with the wrong drug? I'm just trying to visualize.
So very specifically, the drug that we'd want to select, right? If we plug it in our digital twin, it would change the time of labor by several weeks, like decelerate it or delay it by several weeks, based on measuring these immune response. At the same time, we wouldn't want to affect how Tregs are being Treg function, how Tregs are being responding during pregnancy, because we know that if you give a drug during pregnancy, around 24 weeks of pregnancy, maintenance of pregnancy is really dependent on Treg function. So you wouldn't want to have a drug that by mistake is also really altering how Tregs are functional.
And so what would happen to Tregs if they were altered? The woman would actually just give birth early, so it would be moot. The benefit of the drug would be just counter.
Exactly. Yeah, you would.
It just wouldn't work.
You would risk to have serious side effect or unwanted effect. You'd want to be as selective as possible.
So I'm guessing that there is a limited number of drugs that you're testing, and you've got this assay, and you've got your digital twin. Clearly, this is hard work, but it sounds like maybe it could be you could have a bingo answer kind of quickly. Or is this much more difficult than I'm making it sound to be?
Well, I think we're getting close. I think this assay is going to really accelerate. It's designed to accelerate this whole process, right? So, and this idea of digital twin is also designed to accelerate the next step, which is once we have, let's say, 3 or 4, most promising drug candidate, then we want to be able to test them in clinical trials. In order for clinical trials to be efficient, more and more these clinical trials are driven by biomarkers, driven by surrogate biological outcomes, so that you can limit the number of patients that are enrolled and have data, relevant data in a much faster time point, and just accelerate the whole clinical trial development process. So all this homework that we're doing right now is designed to accelerate the next phase, which is the most difficult phase. Is how do you translate knowledge that is done in the lab into an industry type of a paradigm where you are making the leap and then having eventually in the hands of the physicians and the patients.
So you think you're close to choosing the drug and starting the background work for the clinical trial?
Yeah, I think we're close.
Do you have a candidate already in mind for the drug?
So there
or maybe a couple contenders.
Yeah, I mean there's a whole story that's being developed around lansoprazole that I think Marina must have talked about, and she's talking about it at length because there are many evidence now from both studies in her in silico screen, our drug screen, study from Sing Wei's lab and David Stevenson's lab that are all pointing towards, perhaps that's a drug that would be useful in preventing labor. But that's only, very early stage and only one of the various candidates that we hope to get, within the year, really, when we're done with our first screen.
Super exciting. And you're also going to validate your model outside of these clinical trials. You're going to continue to validate the actual machine learning model. So, when we think about all the next steps from choosing either lansoprazole or another drug, from beginning the clinical trial, from validating the model, all this kind of stuff, when do you think the next big milestone can come from this project and this team, Brice?
Yeah.
When is the next update?
So the big milestone definitely is generalization within other population. We're talking about precision medicine and population level and how important it is to have something that generalized to other demographics, other populations. So we're engaged in several studies that are ongoing with WashU, Nardi Gomez-Lopez, in Wayne State with Adi Tarka. That's in African-American patient population. We have another studies with Jelmer Sprints with a population in Netherlands, another one with Petra Ark and Ines Stelzer in Germany. And so all this data is absolutely critical to make sure that, you know, those biomarkers that seems to be reliable, stable, et cetera, they are actually consistent across demographics and across culture and across geographic regions. And so this is work that has to be done, and that will be the next big follow-up.
And correct me if I'm wrong, but it wouldn't be surprising if some of those biomarkers change based on demographics, right? And that's the whole point of precision medicine, right? That it is not the same for every woman.
Yeah, and by increasing the population and the diversity of the population that we're studying, we only make these algorithms better. We only, you know, are able to then fine tune which biomarker is applicable to which patient.
Wonderful. Well, there's a lot to look forward to, Brice. Please come back when you have an update on any of this. And thanks for your work in this field. It's been really a pleasure having you on, and very informative and super fun.
No, thank you so much for having me. It was a great discussion.